Welcome, everyone. Thank you so much for joining us for another Museum Week program. My name is Carrie Woke, and I'm an educator at the museum. So normally I'm downtown planning the museum's big events like Bugfest, but today I'm here in my living room. For Museum Week, we are focusing on the museum's research and collections section. As a collections-based museum, the Museum of Natural Sciences has thousands of specimens preserved to further scientific research. Today, we are focusing on our herpetology collection and herpetology research. Herpetology is the study of reptiles and amphibians, and we have 300,000 specimens from as far back as the 1850s in our collection. So the museum's collection is the 11th largest in the country and one of the most complete regional collections in the entire United States. So the collection also maintains important collections from elsewhere in the world, including the Southwest United States, Southeast Asia, Central Africa, and Cuba. So today we're going to talk to Dr. Bryan Stuart about the herpetology collection and his herpetology research. So Bryan's the research curator of herpetology here at the museum. Most of his research is focused on discovering and describing new species of amphibians and reptiles. He's especially interested in those species that are hard to identify using only morphology, which is what they look like on the outside, but instead require different tools such as DNA sequence data to recognize. So welcome, Bryan.
Thank you, Carrie.
Awesome. So tell us a little bit about your research. I know you travel all over the world studying herps, right?
I do. I'm lucky to be a herpetologist, meaning a biologist who studies amphibians and reptiles. And remember, amphibians are things like salamanders, newts, frogs, and toads, and reptiles like snakes, lizards, turtles, crocodilians. I am very lucky to be a herpetologist here in the wonderful state of North Carolina. We have a fabulous diversity of amphibians and reptiles. Just a wonderful, wonderful place where we see a mixture of species that are, really characteristic of the southeastern U.S., things like coral snakes and eastern diamondback rattlesnakes that get up into our state, all the way up, and elements of a northern fauna, things like wood frogs and spotted salamanders. So it's a really exciting state to live in. Also, as a herpetologist, I'll just add, North Carolina has more species of salamanders than any other state in the country. So I'm very lucky to be a herpetologist here in North Carolina. But like you said, indeed, I do sometimes travel to places around the world, especially places that have been very poorly studied, historically underexplored for diversity of amphibians and reptiles, meaning what species occur there and where. And so I very frequently do field trips, usually to parts of mainland Southeast Asia, like Laos, Cambodia, Thailand, Vietnam. And I work with my colleagues in those countries, and we do inventories, and we learn what species occur where. And during the course of those inventories, we often make new discoveries, like first time a species has been seen in decades, first time it's been photographed in life, first time it's been known from that country, and very often species that have never before been known to scientists and that my colleagues and I are able to describe as new species to science.
So awesome. So I personally love herpetology, especially snakes. But my next question is, I want to know about your craziest adventure as a research biologist.
Well, I have a couple of really fun, almost every field trip results in some exciting moment, when you're in the field and you find that amphibian and reptile, and it's something you haven't seen before, or something you weren't expecting to see, or maybe it's a new species. So I just have countless memories. But I can definitely share with you one or two stories that stick out over the years. Let me put up a picture here behind me. I'm going to move out of the way. This was a moment in the field in eastern Cambodia near the Vietnam border. This would have been in 2003. And it was a wonderful field trip for many reasons, primarily because it was the first time I ever worked with with a fellow named Ying Ti, who is today Cambodia's sole herpetologist. It was the first time he and I had ever been in the field together, and today he's just a superstar in that country. But anyway, on this very first field trip, one night we were doing a survey and we ran into these two snakes. Here's a photograph behind me, and I'll try to walk you through what we're looking at here. Now, so you understand what our inventory work looks like, we conduct it during the day and especially at night, because very often amphibians and reptiles in tropical forested areas will come out and things that we don't see during the day. So in this particular case, we were walking up a forested stream in eastern Cambodia in 2003 with headlamps, and we were searching the streams and the vegetation above the streams for amphibians and reptiles. And my colleague T, who I mentioned just a moment ago, was on the opposite side of the stream. It was about a 15-foot wide stream. And he said, "Bryan, two large snakes." And so I ran over, and these two snakes were on the bank of the stream. So I'll tell you what we're looking at, and I'll tell you that when this happened in real life, I was also struggling to understand what was happening. What this is I'll put my finger up, the snake on the top, the darker one, is a king cobra. And that's about an 11-foot king cobra. And the snake that is in its mouth, it has in the middle of its body, if you can see that, the sort of spotted snake, it has in the middle of the body, the king cobra has it in its mouth, that is something called a keeled rat snake, Tias carinatus. Now, this moment was special to me for many reasons. First of all, it's uncommon to run into a king cobra. And this was a large one, so that was exciting. And king cobras are the world's longest venomous snakes. But they specialize on eating only other snakes, or what we call squamate reptiles. They'll occasionally eat large lizards, but they primarily eat other snakes. And this one was in the process of eating a snake that I couldn't believe I was seeing. That keeled rat snake is that's about a 6.5-foot snake. It's harmless, it's not a venomous snake. The king cobra is a venomous snake, of course. And that keeled rat snake was the first time that species had ever been found in Cambodia. And in fact, the nearest record to it was hundreds of miles away. So we're standing there and we're looking at these two large snakes, and I'm in absolute disbelief because in the mouth of a king cobra is a live keeled rat snake, something we never expected to see. So now I had the challenge of catching both of those snakes. That's an exciting story I can save for you another time, but I will tell you this. As you can see, the king cobra already has that keeled rat snake in its mouth, meaning it's delivering venom through its fangs into the body of the keeled rat snake. At the end of that evening, we released the king cobra and we kept the keeled rat snake. It died during the night in our camp because it had been envenomated by the king cobra as a soon-to-be meal that we interrupted ever so rudely. And we preserved that specimen of that keeled rat snake as a museum voucher specimen. Today, at the time, I was just a graduate student. I was a graduate student at the Field Museum of Natural History in Chicago in the United States. And that specimen is preserved there. That's where I was at the time. But today, that record stands as the only record known from Cambodia. And you can still see the bite marks of the king cobra on the preserved specimen. And all these years later, we're going on 17 years now, the species still hasn't turned up in the country. So it's an important record, and it was just a really exciting and thrilling moment for so many different reasons.
That is so cool. So ever since I was, like 5 years old, king cobras were my favorite animal because of Rikki Tikki Tavi. So I was the kid who liked the cobras, not the mongoose. So that is an amazing story. So that may answer the next question I have, which is what until now has been your favorite or what you would consider to be your most important discovery. So if those two are different, please tell us both stories.
I'm going to tell you about this animal here over my shoulder. Now, this might look like a very large salamander, but actually this is of course zoomed in. This animal in real life is, about 6 inches long. And this is an animal called the Lao Newt. And the discovery of this species is one that I think will always stay with me for the rest of my career. It was both a thrilling and also a very heartbreaking, but also I think a story that sort of has a happy ending too. And so because it was such a special experience, I'll share it with you as probably one of my most important discoveries. So the Lao Newt is actually the very first new species of amphibian or reptile that I had ever described to science. At the time, I was just a graduate student, like I had mentioned before. And this is a really special salamander that lives only in a very, very small area in a very small part of the Southeast Asian country of Laos. And I first learned of this animal when one of my colleagues, a Lao colleague, had come back from a visit to his hometown in a rural part of Laos to attend a wedding. And while he was at that wedding, some people were serving a traditional medicine alcoholic drink. In other words, they had preserved some salamanders in a bottle of locally brewed rice whiskey. And he thought that they were unusual. And so he brought them back to the capital city and showed them to me, these animals that had been preserved in this Lao whiskey for traditional medicine purposes. And I couldn't believe it. It was, first of all, the first record of any salamander from the entire country of Laos, but it was also morphologically in its appearance completely different from any other known Asian salamander. So my colleagues and I, we set up some plans, a field trip, and we went up to that area where our colleague had attended this wedding, and we did field work, and we found this animal in the field, and we did the very traditional approach. We preserved some specimens, and we described the species as a new species to science in a scientific journal called the Journal of Herpetology. And there we gave it the name, at the time it was called Paramesotryton laoensis, meaning found in Laos. Today it's been moved to its own genus. It's the only species in the genus. A genus is a group of closely related species. But here meaning it's unrelated to any other Asian newt. It's called Laotriton. So its name is Laotriton laoensis, the Lao Newt from Laos. And in any case, so what started out for me as a graduate student as a very traditional approach to discovering and describing new species, where you preserve some voucher specimens in a natural history museum like ours, and then you describe it in a scientific journal, you give it a name, you designate a special specimen, something called a type specimen, which we might have time to talk about in just a moment, and so forth. And then I moved on to the next, because so many of these field trips generate so many new discoveries. There was a lot more to do. And then something very unexpected happened with this animal. People who decided that they wanted to collect very rare species of salamanders as pets wanted this animal. And an illegal trade network began seeking out this animal in the very small area that it was known from in Laos. And it became, a demand came for it in Europe, in Japan, and eventually the United States, to have Lao Newts as pets, because they're such an unusual salamander. They're so rare, they're large, they're warty, they're colorful, and so forth. And what's so unfortunate is that the people who are looking to exploit this population for commercial purposes to sell these animals as pets into the international pet trade use the original scientific description as a roadmap to find the species. And so my colleagues and I we spent a lot of time sort of talking about this challenge of how is it that we can describe new species to science for the purpose of conserving them without inadvertently putting their populations at risk by those who wish to exploit the population for personal gain. And so I've been involved in a lot of conversations and publications and so forth dealing with that issue. So what started out as just a straightforward species description became a really important discussion, I think, in my field on how we taxonomists, people who discover and describe new species, can continue to do so because it's so important, but in a responsible manner. I will then add, on a more positive note, to finish up the Lao Newt story, it was quite clear that we needed a lot more data on how much illegal trade was going on in Laos and how geographically widespread and common was the species. And so in 2012, I teamed up, I found a master's student in Laos who wanted to study this for her master's thesis. And today, her name is Somputwan Kimachak. She became Laos's first herpetologist. Today, she's a professor at the National University of Laos. She and her students continue to study this species and other salamanders that have since been found in Laos. And as a result of her efforts, she was able to get this species to become the first amphibian species to be formally protected by law from trade in the country of Laos. So bringing it around full circle, fortunately, the species now has protection. Unfortunately, illegal trade does still continue, but at least now there's some mechanisms for enforcement.
That's an amazing story, Brian. And I think it brings up something that a lot of people don't realize, that so many reptiles and amphibians are at risk because of this pet trade. So is there something that we can do to help curtail the exploitation of these species in the wild?
Well, absolutely. I would think you need to be a responsible shopper. You want to make sure that you're not purchasing animals that have been captured from the wild, animals that have been bred in captivity. And if you're going to keep them as a pet, and you want some to be supporting the trade in something where a small, vulnerable population is not being exploited. So just as many people are really, careful, I think, and responsible consumers when, say you go to a seafood market and decide you want to buy fish for dinner, and you might do some research and say, how sustainably harvested is this species? I think we should be expected, if somebody is going to be buying an amphibian and a reptile as a pet, to be responsible enough to understand what is the conservation status of this species. Where is this animal that I'm actually buying to keep in my home coming from? Are there consequences to me purchasing this animal as a pet to the wild populations? Because ultimately we all share the goal of wanting to keep these things out there in the wild. And so being a responsible pet trade owner, I think, is absolutely critical.
All right, awesome. So I have a couple questions. So Harmony wants to confirm. So the salamander was found in Lao whiskey, right?
The first specimens of this species that I had ever seen were specimens that had been preserved in Lao whiskey by local residents in that area to consume the whiskey for medicinal purposes. That's right. And you raise a very important point. Local people in that area were very familiar with this animal. And I think historically, for many, perhaps even centuries, have been harvesting it in very small numbers for traditional medicinal purposes, and probably without significant detriment to the population. But what changed is when there was suddenly an international demand for this thing. Get as many as you can and get them out of Laos and send them around the world. And so it was a very, it was a very different kind of exploitation in terms of scale and scope.
So I have another question. So is the Lao salamander endangered? I know you mentioned it was protected, but is it officially listed as an endangered species?
It is. As of about 5 years ago, it was officially listed as an endangered species on what's called the IUCN Red List of Threatened Species. Absolutely. Because what we've learned, the species is now known from about 14 different localities, but they're all in a very small area. And what we know is that the species only actually occurs not just in a very small area, and not just in specific streams in that small area, but only in specific parts of the streams in that small area. And this species, unfortunately, has an attribute to it that makes it very easy to harvest in large numbers. And that is that they tend to congregate in these clear stream pools at the headwaters of the very few streams that it lives in. and it's active by day. You can see it sort of moving along the stream pool in the bottom, and they congregate in fairly, in groups. And you can see the warty skin on that animal behind me. It has toxins in its skin. And when you handle this animal in life, it exhibits almost like glue-like substance, and that's the toxin. And I think these species have evolved this bright coloration because they're toxic, and so they're very comfortable moving around on this clear, shallow stream pool bottom by day, knowing that they're protected because of their toxic skin. Well, unfortunately, the fact that they congregate in these small, clear stream pools during the day makes them very easy to harvest, out the whole population very quickly. In other words, they're not scattered, over the landscape. So it's just another unfortunate attribute that made it so susceptible or so vulnerable to exploitation by the pet trade. And by the way, before I forget, I pointed out the warty skin on that animal. I don't know if you can see. I gotta see if I can move my finger. Can you see right above my fingertip? Do you see sort of a yellowish, looks like a yellowish wart kind of all by itself in the black area? Are you able to see that?
Yeah, I can see it.
So that is actually, it turns out to be a water mite. And that species of water mite is only known from the Lao Newt.
That is cool.
So it's,
two for one.
Absolutely. So it's sort of interesting because, we're not just talking about discovering a species of salamander that's only known from this very small area in Laos. We're also talking about, on that species, it has its own host that is also found only in that very small area, and so forth. And
for conservation purposes, when you're conserving the salamander, the newt, you're also conserving its specific mite as well. So again, 2 for 1 in both cases. So I've got a question from Alex. So it's a long one. So here we go. So what are the standards in herpetology on whether a new animal is categorized as a new genus or just a new species of an established genus? So is it determined by morphology? So this is going to your research interest, Bryan.
I'm going to center myself. That's a very interesting question. And there's a lot of disagreement among taxonomists about that. Where there is agreement is almost any biologist that I know would believe that a genus or the species that are within a genus should be a natural group. In other words, they are all each other's closest relatives. In other words, you would not want to have a genus where you have, say, 3 species in the genus, but one of those species is not the closest relative to the other 2. So we all agree that when we assign a taxonomic name, a level of classification to a group of organisms, that we want all the members of that group to be each other's closest relatives. We call that a monophyletic group. However, where it gets very challenging is when you have a phylogeny, a tree of life, is determining how deep back into that tree should be a genus. How deep back into that tree should be a family. And that's where you tend to get a lot of disagreement, and that's why you often will see alternative classifications for particular groups of organisms. So sometimes you might see, the scientific name for our American bullfrog, Rana catesbiana. And sometimes you might see the name Lithobates catesbianus. Okay, and you're wondering, why on earth can't herpetologists agree on which name to use? What we know is you've got this tree of life of these frogs, okay? And we know these things are closely related, but there are groups within that group called the Ranidae, or the true pond frogs. And where you decide we should, call just this natural group the genus Rana, or push it back deeper into the tree, is where you get into the disagreement. So to summarize, I would say we all agree that a genus, a family, so forth, these all need to be each other's closest relatives. But how deep back is really somewhat subjective. And then you asked about morphology. I will just tell you that as we try to figure out evolutionary relationships among species and figure out then how to apply a classification system to them, we use a whole variety of data sets. Certainly molecular data like DNA sequence data are very, very, very popular and very commonly used today for figuring out groupings, but morphology is still used, and sometimes a combination of both. And you can imagine situations where you have almost no choice to use one data set or the other. For example, if you're trying to figure out the evolutionary relationships of extinct things like a group of dinosaurs, well, it's going to be very difficult to use DNA sequence data to figure out those evolutionary relationships. And likewise, a group of organisms like bacteria, for example, or some other microorganisms where their morphology is just so similar, their external morphology, but they're genetically different. And you can't get a whole lot of information out of their morphology. So there you might rely very heavily on genetic data for figuring out species boundaries and groups that you might group into genera and families and higher level taxa. So.
Very cool. So now let's switch to the collections. So I know that you, your office and the collections are not on site at the museum downtown. So tell us a little bit about our research facility and what we have there and why.
Yeah, I'm going to take you into my very favorite place at the Museum of Natural Sciences. As you all know, with so many wonderful parts of that museum, but this is my favorite. And this is a part that many people in the public have not been able to see. This is a part of the museum that's, for safety purposes, is closed to the public. This is a working laboratory. We call it the Research Lab, and it's a very special facility that is specially designed for housing fluid preserved natural history specimens, meaning those specimens that are traditionally primarily maintained in ethyl alcohol and or formalin, and sometimes some other alcoholic preservatives. This is a facility that is located about 6.5 miles away from the downtown museum that we all love and know so well. It's out near the museum's Prairie Ridge Eco Station, which I encourage you to visit if you've never been out there. But this part of the museum, this facility called the Research Lab, is closed to the public because it is filled with so many invaluable natural history museum specimens that are preserved mostly in fluid preservatives that present some, hazardous material, hazmat requirements, and it's a working laboratory. And so we, for safety reasons, it's closed to the public, although we do give scheduled tours when able. And out there at that research lab, there are 4 different collections, and one of those is the amphibian and reptile collection. And the amphibian and reptile collection is what you're seeing behind me. So on, let's see, so on above my left shoulder, everything down, so above, so right over here, look, my arm, there we go. Everything over on this side of the room are the amphibians. So we have the salamanders, the frogs, and another group that's unfamiliar to a lot of people called caecilians, which we don't have any here in the United States, but they're a limbless, burrowing, or aquatic amphibian. They look very much like eels. And then on the other side of the room, over my right shoulder, we have the reptiles. And so these are grouped by crocodilians, turtles, lizards, and snakes. And so you can see it's a big facility, and we have a very large and important preserved collection of natural history specimens of amphibians and reptiles that go back to the mid-1800s.
Very, very cool. So we have another question. So Harmony wants to know if reptiles and amphibians are poisonous.
Good question. Herpetologists love that question. So, the term poisonous is so frequently applied to what a herpetologist would call a venomous snake. So to clear up any misconceptions between a poison and a venom, a poison is a toxin that you ingest, that you would eat, whereas a venom is a toxin that is injected. So a snake, a snake that has the ability to deliver toxins, for example, a rattlesnake, is a venomous animal. but something that you would have to consume to ingest the toxin is poisonous. So frogs secrete poisons from their skin, and so the toxins, in order to get toxins from the skin of a frog, you would have to ingest it or eat it. And so a frog would be poisonous, but a snake would be venomous. A mushroom would be poisonous, but a bee would be venomous, and so forth. Awesome. And then I'll just add one thing. There is one very small exception to what I just said, and that pertains to actually the part of the world we were talking about earlier, in Southeast Asia. There is one group of snakes that have developed these really interesting glands on their neck. They're called nuchal glands, and they are able to secrete a toxin that's a very bad tasting toxin. So we actually have an example of one group of snakes that actually could technically be argued is slightly poisonous because you can ingest the toxin by eating them, but that's very unusual.
Very cool. Okay, so I have a question. What is the coolest specimen we have in the collection?
Coolest specimen in the collection. Well, I can't resist pointing out this part of our collection. These are definitely the coolest specimens in our collection. We keep these at minus 115 Fahrenheit. So these are our tissue samples, our genetic resources that we store in our ultra cold freezers at minus 80 Celsius or minus 115 Fahrenheit. So as we talked about earlier, as scientists try to figure out evolutionary relationships among species, and also trying to figure out differences among populations to decide what might be one species and what might be another, we try to make use of as much data as we can. And today, with the advent of DNA sequencing technology and how it's available it is to all of us, having preserved material from which we can obtain DNA sequence data from our collections has become a super high priority for us at the museum. And so now very often, almost always, whenever we have a new specimen coming into the collection for whatever reason, we'll want to pull a part of that specimen. In the case of amphibians and reptiles, it might be liver or muscle or something like that. And we often will preserve it in a special tissue buffer. And then we store it in these ultra cold freezers at minus 80 Celsius to try to slow down any enzymatic activity and to preserve that tissue for high quality DNA indefinitely into the future. And so today, as researchers at the museum and researchers outside of the museum use our collections, they'll use the preserved voucher specimens, like I've sort of been, we've sort of hinted at, things like this that we'll talk about in just a moment, but they'll also make use of samples like this. And so what we're actually looking at here is, and that's my hand, I've pulled out a tube, and so there we've got some ultra cold frozen liver tissue, and the remainder of the animal would have been preserved in formalin, fixed in formalin, and then preserved in 70% ethanol, is out here somewhere in the range. So now we have the ability of looking at both the morphology of the specimen and also its DNA. And so, as you can see, here's a sort of a close-up of a jar of preserved salamanders. You can see that tag over my shoulder. That's a unique number called a catalog number that's assigned to that individual salamander. And so that number will stay with the preserved part of the specimen, and it'll also stay here with the preserved tissue for molecular analysis. So that we could, because our goal really here is to maximize the utility of these collections, to get as much information and to make them as useful as we can now and into the future. And so I can't resist telling you that these are our coolest specimens.
They are very cool. All right, so I've got another question from Glenn. So are any skin absorbable substances from snakes, are there any skin absorbable substances from snakes, et cetera, and would they be called venoms or poisons? So if you absorb them through your skin rather than ingesting.
No, not that I'm aware of. As we know, those of us who love to catch snakes, snakes will often musk, meaning they'll sort of ooze this this oozy, smelly goo from some glands down near their vent, and it's smelly, nasty stuff, and it's meant to sort of turn you off as a predator that was looking for a meal. That won't intoxicate you in any way. I mean, by getting it on your skin, it isn't going to poison you in any way. But you'll be able to smell it on your hands for a little while.
Yes, I think a lot of us have been musked before. My next question is, what do you consider to be the most scientifically important specimen that we have in the collection or group of specimens?
These specimens over my shoulder are probably, this group, I would say, are probably our most important specimens. And I would argue the counterparts of these specimens at other natural history museums are probably the most important specimens. These are what we call type specimens. Now, this is not a very exciting picture. And I'm sure when people are hoping for, one of the most important specimens, you're looking for something, big and flashy. And instead what I'm doing is I'm just sort of showing you, maybe I'll back up out of the way. I'm showing you the inside of a 700-pound cement-lined fireproof safe. And this is what we call our type specimen cabinet. And why are we so concerned about the safety of these specimens? They must be important, right? Now, I do want to add, just to back up, as we go back to this collection, we take safety in this collection extremely seriously. Because we have hazardous material, we have flammable ethyl alcohol and so forth, we have all sorts of safety measures in place. Fire safety, we have alcohol sensors and so forth. So we are always uber careful with these invaluable and irreplaceable specimens. But these are so important, they go into the 700-pound cement-lined fireproof safe. Type specimens, and this is our type specimen collection of amphibians and reptiles behind us, are the original specimens that are used when a new species is described to science. And so those are actually the name-bearing specimens. So anytime we describe a new species, we designate one specimen, we call that the holotype, to be the name-bearing specimen. And so if I'm going to describe this species A, my concept of what I mean by that species is what is in this specimen, this holotype that I have designated. Behind us are holotypes and some other specimens, other kinds of type specimens, things called neotypes and paratypes and so forth. And we try to keep these so safe because they're irreplaceable. They are the standard for the species. And you think, well, why is that so important? If you lost it, you would just go out and get another one. Well, often you can't, but more importantly, what we learn, especially as we incorporate new molecular tools like DNA sequencing, is that often what we consider to be one geographically widespread species might actually consist of two or more cryptic species. And if we're going to take species A and we're going to split it up into two species, species A and species B, before we can figure out what the new species is, we need to make sure we really understand what was the originally described thing that we're splitting the new species out of. And that's not always an easy thing to do, because very often when species were described, back in the late 1700s or 1800s, people were very vague about the locality or maybe vague about details of that specimen. And today we have to try to reconstruct exactly what that species was described on. And more tragically, I'll add, and more recently, during World War II, some incredibly important natural history, relative to the tragedies of World War II, this is small scale, but within the world of natural history museums, a number of really important natural history collections were destroyed during World War II from bombing. And most of the early type specimens were housed in many of these Western European collections. And a lot of type specimens were lost to the world during World War II from bombings and other tragic consequences of the war. There are all kinds of wonderful stories, just really heartwarming stories about curators who just went way beyond the call of duty and grabbed type specimens during air raids, and a lot of specimens were hidden away in things, underground caverns and things like that to try to protect them from bombing raids and things like that. All kinds of really heroic efforts by museum curators, but a lot was lost. And today, all these years later, taxonomists like myself are still often trying to reconstruct what was originally meant when that species was described, now that we no longer have the type specimen.
Very, very cool. And it's so cool. I didn't realize we had so many. So that is pretty awesome to have in our collection. So I have a question for you. What is your favorite specimen in the collection? You can say one of your favorites. I always have trouble narrowing it down personally.
Yeah, I mean, there are just so many. I have to say, usually my favorites are specimens that are either tied to a story that's very personal to me, there was some moment, or I would call them favorites because they're just so important to the scientific community and just to our natural heritage in general. So let me just show you a couple of our really, really fun ones. Or I would say really, really significant ones that I would call favorites, whereas if I only had a few minutes to show you something in our collection, these are probably the things I would show you. So Carrie, if you could pull up the Neuse River water dog.
I will do it.
This is what's over my shoulder, but maybe Carrie can show it to you a little bit.
Mm-hmm.
A little better way. Well, anyway, what I'll just tell you is this is a species that I think is really is significant to me because of where I live here in the Research Triangle region of central North Carolina, but also because it has such an important connection to the history of our museum, the North Carolina Museum of Natural Sciences. This is the Neuse River water dog. It's a large aquatic salamander that's only found in the Neuse and Tar River drainages here in North Carolina. And it was originally described to science in the 1920s, like this was 1928, the 1920s, by our museum founder, C.S. Brimley. And he named the species Necturus lewisi, and the scientific epithet, the species name lewisi, the I on the end means belonging to a man. So this name means, in Latin, belonging to a man named Lewis. Well, Lewis was a fisherman in Raleigh who was fishing in the Neuse River near to Raleigh, and caught some of these animals and brought them in to Brimley in the 1920s, our museum founder, and said, "What are these?" And Brimley realized these are water dogs, which is a salamander, despite the name water dog, that are totally different in morphology. Brimley didn't have access to DNA sequence data then. Totally different in morphology in their size and coloration and spotting than to other water dogs. And so he named it as a new species. So I just find these to be really amazing specimens because they are really one of our almost backyard endemics. I mean, they're endemic to North Carolina. They're endemic to mostly central North Carolina down into the coast. But it's really our backyard special animal, named after a Raleigh fisherman who brought some of these things into our museum founder, the Neuse River water dog. So that's a really fun one. And then there are some that are a little bit sadder, but just as important. I don't know, Carrie, are you able to pull up the river frog?
Yep. There we go. I got the frog.
Okay. Well, this is what's over my photo. I'm not sure if you can see it any better, but this is something called a river frog, Rana hexturi, or Lithobates hexturi. And river frogs, they're an example of what I started out telling you at the very beginning of our conversation today, are an example of a real southeastern U.S. species that's better known as we get further down into South Carolina, Georgia, Florida, and Alabama, and so forth. And historically, river frogs just marginally got into our state. And we have a total of 14 preserved specimens of river frogs from the state of North Carolina. There are additionally some preserved tadpoles as well. So there's something like 21 preserved records known from the state of North Carolina. The last river frog, these were found down in the Lumber River drainage. So, and so just down in the real southeastern corner of our state, near to South Carolina. The last specimen of a river frog ever verified in the state of North Carolina was in June of 1975. The species has become extirpated from our state. Now it still persists. It's not doing great, but it's still in South Carolina, Georgia, Florida, and so forth. But we have not seen it in our state, for what are we going on now? Well, 45 years. Over 40 years anyway. And so we have in our collection these preserved specimens from prior to 1975 that serve as irreplaceable material to prove that river frogs were once part of our natural ecosystem. Why they disappeared is not clear. There have been hypotheses that perhaps disease, like some of the chytrid fungi that are now known to have detrimental effects on amphibians, might have played a role, but so far there seems to be no evidence. And in fact, these actual specimens of our river frogs from North Carolina that are preserved in our collection have been studied to see if they were infected in a serious manner by chytrid fungi by doing what's called histopathology of the skin. And it does not appear that that was a major problem, at least for the preserved specimens that we have.
So Bryan, I have a question. Is there a plan for any type of reintroduction of river frogs?
It's a very good question. The first thing we need to do is be sure that there are none still here. And we also need to see how close, in like the Lumber River drainage in South Carolina, they get to our state today. And that I think will help guide some of the decisions. And then of course, the other thing is we really need to explore more, trying to understand what might have caused that die-off. Because if there's something out there in the environment, if we put them back, perhaps it would be a futile effort. But what is really positive about our collections here is we know exactly, for the most part, exactly where these things were in our state because of these historical museum specimens. And so if reintroduction efforts ever were to happen, say to move animals from populations in a careful and controlled manner from, say, neighboring South Carolina back into North Carolina, we would have a good idea of where they historically occurred, so we'd be putting them back into a place where they would be a natural part of that landscape.
Very cool. Okay, so I'm going to stop sharing the screen now. So I have a question from Facebook. Did a lot of collections get destroyed in the Great War also? So World War I.
Yes, there was absolutely, and of course a lot of collections got lost, just from other historical events beyond war. But yes, specimens were lost. But I think because of the significance of the aerial bombing campaigns during World War II, and especially on major cities like London and Berlin and so forth, that it's the World War II is really what caused so many painful losses of type specimens that are so invaluable to the scientific community.
So I've got one more question. So Miranda wants to know that she knows box turtles have home ranges, but do snakes have home ranges as well?
Absolutely they do. And in fact, one of my colleagues, another herpetologist here at the museum named Jeff Bean, he's our collections manager of herpetology. One of his primary foci in his research is telemonitoring particular snakes that are of significant conservation concern to us here in North Carolina. These are species that occur down in the Sandhills region of North Carolina. And he inserts these transmitters into these snakes, and he follows these individuals around for as long as he can. In some cases, that goes on for years. And by compiling data like that, you realize that these individuals do have home ranges. Absolutely. They have places they like to go. They have patterns, places they might tend to like to spend the winter, places they might like to go for nesting and so forth. So I would definitely encourage you to direct questions about home ranges and snakes in North Carolina to Jeff Bean, who has great expertise in that.
Right. And some of the things, I remember talking to him, and one of the things is that they not only have like a range fidelity, but like actual stump fidelity, right? So there's like this one stump, and that's where they live, right? And so, so I guess that is, when you're talking about conservation, you realize how important some of these habitats are, and things that we don't, we wouldn't consider a stump very valuable, but these snakes absolutely rely on these things.
Absolutely.
All right, so my last question is, so I just want to kind of focus a bit. I know we've talked a lot about conservation, which is a topic that I feel really passionate about, but how are these collections and research, what is their importance for conservation science? Like, why do we have, you really talked about this a lot, but I just kind of wanted to wrap it all up. Why do we have these collections? Why are they important? Why should everybody listening today support this?
Yeah, our natural history collections are an irreplaceable, invaluable resource. And what these collections have done is they have documented where species used to occur, where they occur today, and they're often used to project where species might occur in the future, say under different climate scenarios. And also almost all aspects of natural history are learned through study of natural history specimens. We use these specimens as just invaluable sources for learning about diet and growth rate and reproductive biology, things that are very, very difficult to observe in nature, but that we can go back through this preserved material, and including looking at series over time and over different localities, and learn information that you can't get any other way. And you can imagine, for example, without these natural history collections, how difficult it would be to really understand what our landscape looked like prior to, our species modifying and disrupting it so much. So for example, we talked about those river frogs. Without these records, without these preserved specimens, it might just be an observation from the distant past that somebody might have put into a field notebook and so forth. But did they really identify that correctly? Sometimes river frogs can be confused in their external appearance with even our common American bullfrog and so forth. We have the specimens, and there's just no way to go back into that lumber river drainage prior to 1975 and reproduce that today. So they're absolutely invaluable resource. What's so amazing about these collections, being a curator, I thought that I had a really good understanding of how of all the potential uses for natural history collections. But we, as you may know, and I should address, these collections are here to serve the community at large. And so we are constantly receiving requests to use the collections, either for people to come and examine the specimens themselves or receive images from the specimens, or to receive loans of the specimens. We're shipping specimens, including whole voucher specimens and little pieces of DNA from our ultra-cold freezers that I mentioned, all over the world for people to do research on our material as well. And it would just, every time I think that the potential uses for these collections have been exhausted, we get a request to use the collections in a new way, in a new and exciting way. We have artists requesting specimens. Are these specimens served as such important components of things like field guides and so forth? There's all kinds of incredible needs for these collections. And so because they're historical, we just can't, they represent a species composition and distribution from the past that doesn't look like it does today because of what our species has done to the landscape. there's no way to reproduce that, to replace this. And so it's absolutely a critically important historical treasure to our natural heritage.
Well, Bryan, that was such a fantastic presentation. I love how you bring your passion for these animals and the collections to all of our visitors and our listeners. I love our collections. I love the museum. And I just think that they're such an integral part of what we do every day. So I really appreciate you joining us today. And to wrap up, that is the end of Museum Week. So this was our last presentation. Thank you so much if you've joined us all week. It's been such a pleasure sharing this part of our museum with you that isn't often highlighted. It's behind the scenes. You come and visit, you don't get to see it. So thank you so much, Bryan. Thank you, all of you, for joining us. And we have lots of other programs going on, virtual programs right now. So please check our website, tune in. I know there were some questions we didn't get to, so feel free to email Bryan or I with your questions. We're both easy to find on the website with the staff email. And we will see you soon. Thank you.